Now the serological methods will be continued after the agglutination and precipitation test. We will see the complement fixation test, which is also called CFT. Its principle is the complement is an immunological test used to detect the presence of specific antibody. Because its name creates the complement, so specific antibody and antigen on the basis of complement fixation. The complement fixation is a system of serum proteins that react with antigen and antibody complexes. Therefore, it is called the complement fixation, and if the reactions occur on the cell surface, it will result in the formation of transmembrane pores and therefore destruction of the cells. On the basis of these transmembrane pores and the destruction of the cells, we will see that whether the complement is fixed or not. It has different kind of the result interpretations in the step 1, step 2 and step 3. We will see it in detail in later and how it works. The viruses acts as the antigens in the complement fixation test. The virus antigen for this test obtained from the infected animal tissues. Of course, it is one of the disadvantages of this test, just like the agglutination and precipitation test, that we have to use the animals for isolation of the viruses. However, in this the antigen antibody test, the complement fixation test, similar way the virus need to be used, and virus is obtained from the animal tissues, and the tissues may be it depends on the which kind of virus is this. Most of the time we are using the lungs, or maybe we are using the brain, just like for the rabies virus. Then the application of the complement fixation test, it is used for the detection of specific antibodies in the serum. What kind of the antibodies are present in the serum, it will be used the complement. We will use the complement to fix it and see identification of the unknown virus by using the known antibody. Similar way, just like the agglutination and precipitation test, here we have seen the antigen and the complement which is fixed with the body and the serum without antibody. So most of we can use the control, and we can use it on the both phase, and we can see that whether the complement has been fixed or not. The little procedure about the detail, the serum is separated from the patient, and then patients are naturally have different levels of complement proteins in their serum. We can use different types of proteins to indicate the effects of this might have test on the complement proteins. Therefore, we will see that which kind of the proteins is needed and which kind of the proteins are need to be used for the complement fixation. So, if we know the known amount of the protein, we will use that protein, and if we know that we don't know the virus, we will detect the viruses. The serum is heated in such a way that all the complement proteins but not the antibodies within it are destroyed. A known amount of the standard proteins are then added to the serum, and these proteins are frequently obtained from the guinea pigs, as we already know that we have to use the animal body for the preparation of the antigen, which are the viruses. So most of the time we are using the guinea pigs. The antigen of interest is added to the serum, and then the sheep red blood cells which have been pre-bound to anti-SRBC antibodies are then added to the preheated serum and foresee the results presentation. If the results interpretation which are included in the step 3, step 2 and step 4, and we will see the different results. If the patient serum contain the antibodies and thus we have used the antigen, the similar antibodies are present in the patient serum, then they will to bind into the antigen, and on the step 3 we can see that the complement has been fixed with that antibody. However, if the complement proteins will react these complexes and will be depleted. Similarly, if the red blood cells serum antibody complexes are added in step 4, it means that it has continued toward the step 4, which is no complement left in the serum. If no antibodies against the antigen of interest are present, the complement will not be depleted and it will react on the SRBC antibody complexes, which we are using in the step 4, analyzing the SRBCs and spilling their contents into the solution, and the solution will be turned towards the pink. If the solution is turned towards the pink, then it means that it has gone to the step 4 and the complement has not been fixed with the antigen, and there is no reaction with the antigen and antibody complexes. Just to understand a little bit better, we can just see a little video here